Welcome to PHA 20034. We have here some nice slides that introduce the topic of hair structure. Now, we also have some videos here that also introduce some of the products that are associated with hair and also some very basic overview of hair. Now, we'll explore this as we get a bit further, but please watch the videos and enjoy them. Now, I've titled this slide Learning Objectives. Really what we are going to be looking towards is the physiology of hair, the protein structure and the chemical bonding that's in there, because that cascades knowledge down to all these other sides of the science to support further components such as development, formulation, safety, regulation, manufacture, sustainability. All of that is very, very heavily dependent upon understanding the physiology, the protein structure, and the chemical bonding that exists in hair. Once we've cracked those other subjects become much simpler to understand. We'll have another look at the structure of hair at a very top level here. If we look towards the integumentary system, which we've learned about in the first year, we see that we have hair growing out of the skin, the hair shaft. That's very easy, very visible. What we have is the actual structure that contributes to that is below the epidermis, and we have associated with that an epicrine gland secreting fluid, and we'll talk a lot more about that as we move forward. But the main thing that we really want to start saying is that we have a lot of detail in this structure, especially around the root of the hair. Now, one of the key things that we didn't see in that previous slide was the erector pili muscle, and that actually pulls the hair up straight and gives you, is what's responsible for giving you the goosebumps, and that's an important reaction to cold, so that you can actually create a layer of warmth around you by using your hairs to actually stand up straight and make it more difficult for air to move across your skin. Okay. During the anagen phase of hair growth, several key physiological processes occur to actually grow the hair. First one I'm going to talk about is keratinization. Now, some of that will be handled a little bit later as we talk about the structure of keratin, but what's going on at the moment in this phase is you're getting this production of keratin, which is the hair protein, and it's that hair protein that actually gives the rigidity and the strength to the hair. That's the really functional part of what hair is and what we understand by hair. And of course that's built up by amino acids being put together, and that requires a lot of energy, because putting amino acids together, they don't want to do that. You actually need to actually put in energy to make that occur. So there's an energy requirement, which means there'll be a blood requirement, and what you see is a rich vascular network around this bottom of the hair, and that's really because you've got not only energy that's needed in terms of oxygen, sugars, you've also got nutrients to supply to make sure that the enzymes can function that can do that. So there's a broad range of not just amino acids but minerals that will also be needed to actually produce that hair, because most enzymes have a mineral at the core, and that's what actually makes amino acids join together. Now, what you've also got going on here is you've got your melanocytes, and the melanocytes are generating melanin as the name suggests, and that's what gives the hair its colour. And again, they're part of this dermal pili, and you'll find them at this point in the hair root itself. The result is you get a hair shaft elongation. The hair actually grows, and it'll grow for a long time. You know, you've got this 3 to 5 years for the hair to grow on your head, and most of your hair on your head is typically in this phase. And I'm going to move now to the next slide. The catagenic phase is a transition phase towards the resting of the hair. And what we're seeing is this movement down of anything that's associated with that hair growth. So what we're going to see is a reduction in the cell matrix activity. So we're going to see our matrix activity starts reducing around here. So we're getting less cell division or mitotic activity within the bulge region of the hair follicle. We're going to see a decline in the proliferation of any signaling that's occurring there with hormones, and so we're going to actually see a reduction in the blood supply as well, so that rich vascular network is lost, and what that means is you're getting a starvation of nutrients and energy, so everything you need to actually make hair is just less around, and so subsequently you've got a downgrading or less keratinization occurring. The melanocytes are not producing the colour that you need for the hair as well. So this is all a reduction in all the structural components that are going into making hair. Essentially, that's what's happening, and the follicle starts to shrink as the growth phase concludes, and you get a sort of miniaturization going on, and it's associated again with reduction in metabolic activity and of course low blood supply going on there, and eventually you get hair detachment and hair clump formation. The hair shaft detaches from the dermal pili, this bit at the bottom, okay, and due to the disruption of the epithelial connections at the base of the follicle, you see the loss of the actual hair itself. This detachment of the hair strands, well, begins the transition into a club hair, which is characterized by a distinctive club shape at the end, and that remains anchored within the follicle until the telogenic phase. Until the tenogenic phase, when it is eventually shed. The telogenic phase represents the final stage of the hair growth cycle. The primary function of the telogenic phase is to release the hair club formed during the catagenic phase. The epithelial attachments between the inner root sheath and the hair shaft degrade under this time, and it's this degradation that allows for the detachment of the hair from the follicle. This shedding process occurs naturally, and you'll have about 50 to 100 hairs lost daily. What we also have is a process known as follicle involution. This is a reduction in the size and also a change in the metabolic activity that's going on. It's a reduction in the metabolic activity. Your blood supply becomes absolutely minimal, and the follicle enters a state of dormancy. You've basically got a sort of phase of resting and renewal while the hair growth ceases during the telogenic phase, and the follicle is just entirely inactive. The dermal pili located at the base of the follicle remain functional, and it maintains contact with the bulge region. And this connection is crucial for the next phase that you're going to see, where the actual growth is restarted. Exogenic phase. The exogenic phase is really the final act in this process, and sometimes some sources actually leave this phase out. So I hope that helps in sort of trying to say how important this phase is and how useful it is in terms of trying to describe this process of hair growth. But in essence, what you've got is, if the hair's in the shaft still, it'll be released. It's essentially what we're going to say in this is the key to this process. But it's a bit more than that, because what's going to be happening is that you've got this minimum follicle activity. The hair follicle's pretty inactive during this point, with minimal metabolic activity and blood supply. But during this period, it starts to prepare for growth. So although the hair's shed, the follicle's not entirely dormant, and what you get is the surrounding dermal cells will continue to prepare the follicle for the next growth phase. We have to think that, though, that this phase is essentially very short, and lasting only a few days to a few weeks. And what's happening is, following this phase, it's going to re-enter the anogenic phase, where it's actually going to start growth. Now, one of the things that we really do need to sort of draw to your attention is you have actually got stem cells in here, and those are what we're actually seeing here, which are actually going to start this whole process off again, and that's really a key to this. So if ever you get biologists looking for stem cells, tell them about this particular phase of the hair cycle and how they can go and harvest them. Let's talk about the hair physiology, and I think it might be easier just to actually start talking about this going from the outside in. And on the outside we have what's called the cuticle, and the cuticle is made of these layers of overlapping scales, and you've almost got to think of it like a roofing tile, where the next tile down actually is halfway out, and the tile underneath that is another halfway out under the previous one, and so you get this stacking occurring. And what this means is that it's actually quite difficult to get past that cuticle. It's designed to be difficult to get past that. It's designed to protect what's in the middle of the hair. This is a protection system, and it's to protect the hair from water loss. In particular, the protein shape will change with water loss. So I mean, we've all sort of heard of hair extending when it's wet, and when it's very dry, contracting. That's very real, and the cuticle is trying to protect the hair from those changes. But you've also got a whole lot of other materials within the hair that it's trying to protect and keep within the hair itself. Now, the medulla, that's the middle section of the hair, and this is everything that you think about with hair in terms of its properties of having strength, having flexibility, having elasticity, and colour. These are all associated with this region of the hair. So And what you've got going on there is these cells that are in there actually contain the keratin, and that's this protein, which is a long chain that actually gives the hair its strength. It's a polymer, just like you might think of a fibre. And I think that's probably the best way of thinking of this. It's containing the cells that contain the structural aspect of the hair. And the cortex, well, this is a bit of an odd part to the hair. Not entirely understood, but we only see it in sort of really thick hairs for the likes of ourselves in terms of eyebrows and eyelashes. It's loosely packed cells filled with air spaces, and we've got the story of the polar bears having hollow hairs. That's again having this cortex empty or nearly air filled. So this is quite a useful feature for actually trapping heat and making this hair a bit more useful in terms of keeping us warm. The structure of the hair cuticle, I'm going to try and talk about here. The cuticle, as we said before, is the outermost layer of the hair, and it's composed of these overlapping flattened cells, which, if we go to the pointer, let's do that, you can see these ridges on this bigger sort of scale electron microscope picture of a hair, and then this actually is probably a better picture showing really what's happening with these cuticles and how the overlap is occurring. Now, the previous picture we tried to sort of idealize this by showing them almost like roofing tiles, but here it's a much more realistic picture of what they're like. Now, you've also got this, can you see this little shape here, this and here? There's lots of evidence of structure going on in there, and we'll talk about that a little bit further on. What we have to remember is the cuticle is particularly rich in proteins and hardened by keratin, which really contributes to its toughness and aids in protecting the hair cortex that's underneath. Now, the actual cuticle is made up of a number of different layers. The A layer, which is the outermost layer, is densely packed with proteins, and it's actually covalently bound to a whole set of fatty acids, and we'll talk about those a little bit in a moment. We've then got this exocuticle, the middle layer, that consisting of protein meshwork, which is embedded with lots of lipids as well. And then we've got the endocuticle, the innermost layer, which is highly structured, and we talk about it being amorphous, so it doesn't have that regular spiral structure of keratin within there. These are actually proteins which use calcium for their binding and holding them together in a tightly bound structure. So that's somewhat different to what you've seen with what you'll see with keratin structure. Now, the primary function of the hair cuticle is, of course, to protect the inner cortex of the hair from damage, and the sorts of things which damage it are things like UV light, dust. You've got things like ozone in the atmosphere that doesn't help a lot. You've got a lot of chemical treatments such as colouring and perming. These actually damage the cuticle, and some of them really go out to damage the cuticle as a means of delivering their colorants. You've also got mechanical damage such as brushing and combing that'll just you put in a tension on the hair, that can actually damage that. If you actually rip off the cuticle, that again can expose the inner part of the hair. So you've got plenty of things which can actually damage that. Now, what it also does is it reduces the friction between hair strands and makes it more manageable. Now you also see a bit of a shine on hair, and this is partly down to covalently bound fatty acids on the surface of the cuticle, and these actually give it water repellency and make its hair somewhat hydrophobic. You know, it's very easy to get a hair to float on the surface of water, and that's because it's hydrophobic. Now we also want to talk about what we call the epicuticle, which is this membrane structure that surrounds the hair cuticle, and it's thought to be derived from the alayer, and it's believed to play a role in reducing the friction and protecting the underlying cuticle. The cell membrane itself, we talk about the cell membrane complex, and it's a very specialized location between the cuticle cells that help maintain their adhesion and integrity, and it does undergo some changes as hair grows, and this is really what plays an essential part in protecting the hair. We'll look at that in another slide, and we'll stop there for this. Okay, I've called this title of the slide epicuticle, but the epicuticle is just one part of this cell membrane complex, which is actually gluing the layers of the cuticle together. Now, I'd say probably the most interesting part of this for me is these sulfur bonds here between the epicuticle and what you've got as a lipid here, and it's a long hydrocarbon chain there. So that's a very oily like chain, and if you were going to try and attack this, those are very sort of difficult molecules to actually get a reaction out of. The part that actually is most vulnerable is going to be this part around here, the oxygen sulfur bond that bonds onto the epicuticle. So if you're trying to attack this and trying to maybe get a colorant in or trying to change the hair in some way, then this is where you'd actually attack. And it's no surprise that when you actually start treating hair and trying to do something to it, you get a horrible smell, and the horrible smell is usually sulfur being liberated in some way. It forms a lot of very light gases that can be inhaled, and they're very toxic, but in very small quantities, and fortunately they smell horrifically badly at very, very low concentrations. So it may smell horrid, but it doesn't actually mean you're getting poisoned. Okay, and we'll take you back to A-level chemistry here, and we're going to go back to this idea of proteins having structure beyond just the amino acid chain and the sequence of amino acids, and they owe this structure to hydrogen bonding, and keratin is the go-to example of how you have these secondary differences, and it's about where these hydrogen bonds link up to that actually gives it this structure between the alpha helix that you find in hair and the beta sheet that you find in your fingernails. Essentially the same sequence of amino acids, just folded up very differently. And what you're seeing is, where do these hydrogen bonds link to? And within the beta pleated sheet, the hydrogen bonding is quite easy to see between carbonyl groups and amino groups. So carbonyl amino, and so on. So that's just fairly straightforward. Now this is slightly, again, you see the same carbonyl to amine group occurring within the alpha helix. It's simply spun round, and so that you're having that bonding occurring within the strand rather than between two different amino acid chains. So you've got one amino acid chain directed this way, and directed opposite in the beta pleats you have another chain going the opposite direction, whereas in the hair version or the alpha helix you've got the hydrogen bonding occurring within the same amino acid strand. So that's the key takeaway, is the hydrogen bonding is occurring within the same strand rather than between two different strands of amino acids for the beta pleated sheet. You also get this regularity in the bending of the structure. So within the alpha helix you see every 0.54 nanometers you get a repeat. It's almost like curves round and begins again. Within the beta pleating you get this repeating unit every 0.7 nanometers, so it's a little bit longer to repeat. Now I'm going to try and explain that difference between the beta pleated keratin and the alpha helix keratin a little bit further here. Now what you have is the ability for chemical bonds within that amino acid chain to rotate, and you've got the bond associated with the carbonyl group and the amino acid, the amino group. Those actually have a limited amount of movement, so you don't get a lot of movement there. Those will be in a plane, and that's really what the rectangle is trying to represent. But where you actually get that joining together between the amino and the next carbonyl group, you have one carbon atom in between those. The bonds between the amino and the carbon atom can change, so that can rotate through 360 degrees. Also, from this carbon atom to the next carbonyl group, that can also rotate by 360 degrees. So that gives an awful lot of ability to rotate within the keratin chain, so, or any protein chain for that matter. And really what's determining that further is how these side chains to the amino acid behave. And we have groups of these which are very polar. They have OH groups on, for instance, or carboxylic acid groups on, and they can actually want to be water loving, they'll be very hydrophilic, they'll want to be with water, whereas you can also get some very hydrophobic side chains to amino acids, and they will want to basically stick together and stay away from the water. So this repellency and loving of water really is a big driving force to actually creating this secondary structure to the amino acid. You've also got the hydrogen bonding that can occur between the chains, which you saw with the beta pleated keratin, and you've also got ionic bonds that can occur. Now if you have a very highly ionic solution, that can disturb those, and we'll talk about that sometime later. We've talked about hydrogen bonds, but there's also another bond which is a sulfur-sulfur bond that can occur between amino acid chains and within an amino acid chain. Right, this is two slides in one here really. What I'm trying to do here is show there are different types of amino acids that go to make up keratin, and these are the different compositions that you'll find typically in keratin in terms of the amino acid makeup. Now we've got some electrically charged amino acids up at the top here, and the fact that these can ionize means that you can have an electrical charge between two different parts of the amino acid chain, and those can attract one another, or they can within the same chain, or they may attract another chain that's located nearby. So those would actually hold to try and have a force of attraction between those two strands, and that would be via this electrical charge. You've then got some polar molecules here, and these are typically with OH groups and with amide groups and amine groups. So quite a variety of different chemical groupings, and you've then got some special cases which are a little bit different, and then you've got something down here which we talked about earlier. We've got these hydrophobic side chains. Those are water repellent, and those really would want to sort of rest with one another rather than with some of the other more polar side chains. Now if we look to cysteine, that's one of these special amino acids where you've got this SH group, or thiol or sulfur hydrogen grouping, and that can bond with another sulfide, and that will actually cross-link, and so you can get within two components of the same chain. If you see cysteine and cysteine, they can actually bond together, or they can look outside for another chain to bond with, and so you can get a link outside the actual single chain. And again, we've got serine, which is very polar there, glutamic acid, which again ionizes, proline, which has an ionizing group on there. We've got another very polar group, arginine. You've got really quite a lot of very polar groups, really quite at the top here, as well as cysteine. So the hydrogen bonding is really critical to this, and they actually help hold the alpha helix in place and also bond to all other alpha helices nearby. Now I've got another little video that should support this presentation as well, where I sit with some mods and try and talk about structure a little bit further. But this is quite a nice slide in terms of trying to summarize the whole thing. So what we've got going on here, we've got our whole idea of the amino acids sitting as the most fundamental unit of hair in terms of how it's structured. We've then got the keratin protein and those coils and how those are actually connected together as a means of sort of saying, right, that's a next level of structure to this. And then we've got these tetramers that are forming an intermediate filament that are bonded together. And then you've got the microfibrils, which are part of the structure within the actual cell itself, that's part of the hair structure. So you've got this sweeping down from the nano or Armstrong scale through to the micron scale up here, where you've got maybe 60 or so microns across the hair. Now what you've also got here is this structure of the cuticle, and that's always something to bear in mind, that the cuticle is very, very highly structured. I'm going to stop there. Please have a look at the video where I talk about how these amino acids join together, and we'll stop there.